Hello and welcome to the Xapiens Podcast. I'm your host, Loyd Waites. When people think about capturing carbon from the atmosphere, they typically think about planting trees or even entire forests. But the reality is, the majority of carbon that is removed from our atmosphere is actually from phytoplankton and algae that are in our oceans. So, what if instead of focusing on planting trees, it were possible to significantly increase the populations of these algae and phytoplankton? Well, that's the focus of the researcher I have on today. Dr. David Emerson works on iron fertilization. This idea that throughout many of our oceans, there's actually a deficit in iron. So if you fertilize these areas with iron, the phytoplankton are able to grow and have these spectacular blooms that can actually be seen from outer space. And then, as they photosynthesize, remove carbon from the atmosphere. But this process isn't as straightforward as it sounds. Today on the podcast, we discuss many of the difficulties as well as the promises of this research and some of Dr. David Emerson's other research efforts. I hope you enjoy it. Thank you. Anyway, thanks for coming on today. Excited to see you. And so just to start out, would you mind giving just a brief overview of what you work on and what your research is?
Yeah, so I'm a microbiologist, trained as a microbiologist, PhD in microbiology. And I've been working at Bigelow Laboratory for the last 15 years, Bigelow Laboratory for Ocean Sciences in East Boothbay, Maine. So we're pretty well recognized in the sort of oceanography marine world, because we study kind of the basis of the marine food webs in the ocean. So we have about 20 senior research scientists who are principal investigators. We're a small independent research laboratory, so we're not affiliated with the university or have any sort of state funding or anything like that. We're pretty much independent, which gives us a lot of latitude, but also means that we're responsible for raising all our own money. So that's the challenge. And my own background, again, is I was got my PhD in microbiology at Cornell and have worked with metals, manganese, iron, as doing research on those for 30 years or so now. And I guess I got into the ocean stuff through studying iron at hydrothermal vents in the deep ocean and microbes that are using iron, that metabolize iron, live on iron. They get their energy from oxidizing iron. A very unique group of microbes, but they're actually found pretty much everywhere, either in the ocean and hydrothermal vents, sediments, and then in a lot of terrestrial environments as well. So I come into the oceanography world a little bit through the back door, I guess you could say, because my main interest has been through the microbes, but then learned a lot more about oceanography by being at an ocean sciences research facility.
So what do you work on now? I know what attracted me to your work originally was this work that using iron seeding to produce large amounts of algae to try and eliminate CO2. Right. But I know that's only one part of what you work on. So if you could just kind of go through a couple of the projects you think are super interesting.
Yeah, well, that's certainly a project that's very interesting.
Mm-hmm.
And again, I got into that because I study these bacteria that produce iron oxides, mm-hmm. and they produce what we call a biogenic iron oxide, which is a bit different than things that are made in the laboratory or synthesized or mined from iron ores. And they have some unique properties that we think might be useful for an iron fertilization type use. But yeah, my real interest was in the microbes, sort of how they're getting energy from these processes, their ecology, sort of who they're associated with, where they live in the environment, their diversity, those kinds of sort of basic microbiology, microbial ecology, physiology questions. And, we're starting to figure out, I think the diversity things we're starting to figure out. I just came back from the Arctic, where we've been studying iron in the tundra. So the tundra is rusting pretty prolifically up on the north slope of Alaska. And we're nobody's really looked very much at the role of microbes in that process, and their sort of direct role in terms of actually mediating iron cycling. So iron goes between sort of an oxidized and a reduced form, and microbes can do both of those processes. And so, again, it's kind of this interest, I guess, as a microbiologist. I kind of consider myself an organismal microbiologist, that I like to study, sort of whole cells in their environment. We don't really do a lot of hardcore genetics and things like that because we don't have genetic systems for these organisms. So we're limited now. We have genomics, so we can sequence their genomes and learn a lot about them that way, who they are, what they're doing. But it's probably a little bit more holistic type of view. And so how does that lead to iron fertilization?
And what is our iron fertilization for the record?
Yeah, so that's a great question. I guess I can go through a little bit of the history, right, of iron fertilization. So it really developed sort of in the 1980s. So in the ocean, the open ocean, which is the ocean, most of the ocean, right? The part of the ocean that's not close to the continents, has very limiting nutrients, and it's either generally nitrogen or phosphorus, and that's what really controls how much productivity there is. By productivity, it's really the microscopic plants, the phytoplankton that we're talking about, because they're the main sort of primary producers, doing photosynthesis, fixing carbon dioxide in the open ocean. And they're really the basis of the entire microbial food chain. The founder of Bigelow Labs was a guy named Charlie Yinch. He named Bigelow after Henry Bigelow, who was the first director of Woods Hole Oceanographic, who studied the Gulf of Maine pretty extensively and sort of in a holistic way. And there's sort of this saying that came out of that all fish are diatoms or phytoplankton, because that's where everything starts in the ocean, is with those microscopic plants. So getting back to sort of the nutrient limitation question, it was pretty well known that in the open ocean that if you had nitrogen or phosphorus limitation, that's what really controlled how much growth you got. But then people realized that actually iron could be a limiting nutrient as well. And there's an interesting story in that they didn't realize this for quite a while, because they would go out on ships, steel ships, drop water bottles over the side on steel cables, bring the water up and measure how much iron was in it. And it seemed like there was plenty of iron. And it turned out that actually the steel from the ships and the steel from the cables and the bottles and the equipment they were using provided enough iron in that region that made, it showed up on the assays they were doing.
Contaminating all of their measurements by
basically exactly
device.
contaminating their measurements. And so that in part tells you how sensitive things are to iron, because it wasn't very much iron actually, but it was enough to contaminate their measurements. And so then when they started being more careful about how they did those measurements, they realized that actually about a third of the world's ocean is iron limited. And so that came to the idea then. There's a fellow, John Martin, who was a well-known oceanographer, who sort of was one of the fathers, I guess, of developing these idea of iron limitation. So they first, just showing that iron was limiting, and then there's also sort of paleological or paleoceanographic information that iron may have had a relationship to sort of the CO2 levels, especially over the last 400,000 years when we've gone through these periods of glaciation and deglaciation. And there's a correlation, a weaker correlation than there is with carbon dioxide, but there's a correlation with how much iron they estimate was available. And that iron, when you had more iron available, there were potentially more growth of these phytoplankton, which were pulling down carbon dioxide, reducing the total amount of carbon dioxide in the atmosphere, which was then helping to control global temperature. And so then that, coupled with this realization that, iron really was a pretty major limiting nutrient, sort of led to these ideas that, if you went out and added iron to the ocean, you could draw down CO2 from the atmosphere. So that's where this sort of famous quote, and it was really more of a quip, I think, that Martin gave once, that if you give me a half a tanker of iron, I'll give you another ice age. And nobody, and that was, I think, I certainly wasn't present when he gave that seminar, but I think what I've heard is that was, it was very much off the cuff. But it led to a lot of thinking about this process and the possibilities. And this was back in, the 1990s, when among the scientific community, it was pretty much a well-known, pretty well accepted that, climate change was a real, anthropogenically driven climate change was a real thing, and we needed to start thinking seriously about how we were going to deal with it.
Yeah. I always thought it was very interesting because you always hear about planting trees as kind of a way to remove CO2 from the atmosphere, and every once in a while you hear about like CO2 scrubbing.
Yeah.
But if you look at like the per unit area absorption of CO2, phytoplankton blows everything out of the water. No pun intended. And it's not talked about a lot. And so when I dug this up and I ended up finding your research papers on this, it seemed kind of too easy that you can just kind of get this massive amount of iron, dump it into the ocean, and then the phytoplankton, because it's iron limited, will then grow these massive plumes and then start sucking CO2 out of the atmosphere. Why hasn't this been more widely adopted or talked about, do you think?
Yeah, those are really important points first, the really easy part. There's lots of things that start out as really easy, and then you start getting into the details and realizing that, there's a lot more complications there than what people would like to initially believe anyway. But the idea has been, so in the from the 1990s into the early mid-2000s, 2005, 2006, 2007, I think, there were a series of experiments done where they went out with ships and put significant tons of iron into areas that they knew were iron limited. Primarily those were driven by testing this whole idea that iron really was a limiting nutrient. And so in every one of those experiments, they were able to create an algal bloom where you can measure chlorophyll from satellites because of the ocean color. And so they could see these plumes of algae developing. But what they didn't, so that there's two steps to the process. First, you get the bloom, which is the phytoplankton growing, but then they have to sink into the deep ocean. So they're fixing that carbon, but if they don't sink into the deep ocean, then it all just gets recycled in the surface ocean and goes back into the atmosphere within, months to maybe a few years. So the trick is, having this export component to that, where they're getting exported, 500, 1,000, 2,000 meters below the surface. And that's what was not shown by those experiments very effectively, and to some extent it was probably experimental design because maybe the experiments weren't run long enough. Maybe they weren't done in optimal places. But that still led, it definitely showed this iron limitation, and it, through models and stuff like that, people could project that, you could potentially take, gigatons of carbon out of the atmosphere, even on almost a yearly basis. So I can keep going with the history of this.
I have a question about it. I do want to keep going with the history of it. But why is it that the algae has to sink? Because I would think, I mean, in the classic photosynthesis reaction that I learned like in high school, the CO2 gets absorbed and then transferred into glucose. But, I mean, the glucose doesn't create CO2 until it's metabolized. So, So how does the CO2 go back into the atmosphere, even if it just floats around?
Because those phytoplankton, they divide almost every day or 2 days, and then they die. And viruses may actually probably play a significant role in sort of lysing those cells. So you get a bloom, and then it lasts for a few weeks, and then it disperses. And most of those cells end up getting lysed or coming apart, which releases that carbon into the water again. And then there's microbes there that will use that carbon, and we refer to it as mineralization, where it takes it back to CO2, basically CO2 and water, which is the basic building blocks that carbon fixation then fixes that carbon back into sort of biomass. So that's that cycle that happens quite rapidly on the surface. But it's estimated somewhere is around 10% of that carbon that gets fixed in the surface ocean can get into the deeper ocean. And one of the really important questions that we're all struggling with now is how long that carbon, so the ocean's interesting, right? I mean, you look out at the ocean and it's flat and it all looks like it's well mixed, but actually it's got a lot of structure to it. And that's where you get into a lot of pretty complicated physics and stuff like that, because there's different layers of the ocean. And there's sort of this surface layer that's anywhere from 20 to 50 to 100 meters deep, where you have this chemoclines, thermoclines, where you have chemical gradients and thermal gradients that basically separate the deeper ocean from the surface ocean. And if you don't get through that layer, which can be up to 100 meters deep, if the sinking rates aren't fast enough, then everything gets turned over just in that surface layer. And so that CO2 then gets basically immediately returned to the atmosphere. And even if it goes down a few hundred meters, it's not clear how long that CO2 would be sequestered into the ocean. Probably for maybe a few years to maybe a decade, and then as you get deeper and deeper, because this is all happening in the open ocean, which we refer to where the average ocean depth is 4,000 meters, something like that. And once you get down to 3,000 or 4,000 meters, then you're probably on century time scales before that carbon, because there's all these deep currents that are going through the ocean, cycling carbon and nutrients through the deep ocean. And so it's not a permanent solution. Only a small fraction of the carbon actually gets buried in the sediments, and then it's really taken out of the system. The rest of it's sort of getting cycled on different time scales through the ocean. But still, that, if you can sort of take that CO2 that gets fixed and deposited in the ocean and distribute it over its weird equilibration with the atmosphere over, decades or centuries, that's significant. So that's sort of the driving thoughts behind that. But again, yes, I think sort of some of the key unknowns in that are it's really the rate at which that fixed carbon gets recycled back to the surface, what we refer to as ventilation of the carbon dioxide back to the atmosphere. And we don't have, I don't know. I would say we don't have real good handle on that. You might talk to some oceanographers who, there's certainly debate within the oceanographic community about that.
It seems like something very difficult to measure, because, I mean, do you have like CO2 monitors on top of the algal flume? Like how do you see the amount of CO2 that's actually returning back from the flume?
So you can measure the amount of carbon. You can measure the blooms, the actual amount of chlorophyll that's in the ocean, which is proxy for how much algae is there. That can be measured quite well from space, from satellites. And then people go out, and we have automated systems now, remote systems that can measure sort of the amount of particles, the numbers of, essentially, yeah, it's kind of the biomass in the ocean and where it's being distributed. That can be done now pretty well remotely with different. They're basically floats that go up and down in the water column, and then they can communicate with satellites, and so they're autonomous. So you can measure with that. You can get some sense of how big these phytoplankton blooms are, and how much carbon they're potentially fixing, but actually measuring the amount that's getting exported to depth is still pretty challenging. You can do it from a ship pretty well. To do it sort of autonomously is more challenging. And then getting at those ventilation questions, yeah, that's a hard thing to measure, because the thing about, so you've got a vertical component in the ocean, but then you've got currents that are moving laterally. So if you fix carbon in one spot, that will get moved laterally by currents that are deeper in the water column. And I think that becomes quite challenging to how you account for that. And there's very sophisticated models that people have developed, and they're physical models of how these different water bodies move and the currents and how things get transported in the ocean. But yeah, no, those are that's really sort of some of the cutting edge science that needs to be developed.
I feel like you might be able to design some kind of like well-controlled experiment where you have like an algal flume in a box, and then you could kind of use like an RGA, like a residual gas analyzer, to figure out like the composition of the air or something like that, or at least measure the amount of algae that sinks to the bottom of the box. Something like that. Yeah. That's kind of what I'm imagining in my
you could do that. Yeah. And people have done that to some extent. The problem is it becomes, it's a 3 or 4 dimensional problem, right? Because you've got sinking, but then you've also got lateral movement, and then you've got time. And we're interested, one of the things with iron, so if you add iron to the ocean, how long does that iron stay up in the surface where it's available? Because the reason, it's sort of interesting, right? In the ocean, we study iron hydrothermal vents where there's lots and lots of iron. There's, we find these iron mats, these bacteria that are growing on iron, and they can be meters thick on the bottom, on the seafloor, because there's a lot of iron coming out of these vents. There's a lot of iron in the sediments. But because iron forms these oxides that are, basically metallic oxides that are dense, right? They sink. That's what makes iron very limiting in the surface ocean. It just doesn't get mixed up to the surface ocean very well. And so that's sort of primary reason why iron becomes such a limiting nutrient. So yeah, that's one of the interesting things about iron. Is this fact that it's abundant, but in the ocean, because of the circulation of the ocean and stuff, it becomes very limiting. But the other thing about iron and one of the attractions of it for any of these sort of carbon dioxide recovery or capture methods is that you don't need very much of it, because iron, it's used by some of the molecules involved in photosynthesis. It's cytochromes, which are important in sort of our metabolite. A lot of our metabolic enzymes contain iron. But still, the ratio of carbon atoms to iron atoms is something around 16,000 to 1. So in that, purely theoretical terms, you could add 1 atom of iron. If it's what's limiting growth of everything, you can add 1 atom of iron and generate 16,000 atoms of carbon. So when you scale that up, that's where you get this idea that, yeah, we dump a little iron in the ocean and we can get lots and lots of carbon. But it becomes more complex than that in terms of the actual biology of how that works. But still, that is, I mean, we estimate that you add, well, I mean, if you had a couple of bricks here, you could add 2 or 3 bricks of iron to a square kilometer of the ocean, and that would be enough to make that part of the ocean fully iron replete, we say, or not, or allow the phytoplankton and stuff to grow with no iron limitation. So that's the attraction of it, is that you don't need very much.
Yeah, it seems very inexpensive to do something like that.
Yeah, so we've been doing that's one of the things we're doing right now, is sort of trying to do a cost model, a very basic cost model of how much iron you'd need to add to draw down a ton of carbon dioxide. And, you know, depending on your assumptions, it can be anywhere from, you know, under $10 a ton to, you know, several $100s a ton. And that's...
Both of those seem inexpensive. I guess I'm not as familiar with like the climate models, but I mean, a ton of CO2, isn't that like several cars off the road or something like that?
Yeah, I don't know. Yeah. Well, that's a big debate, right? It's what should be the cost of carbon, and that's ultimately what's going to drive a lot of this stuff. I think is how aggressive or how ambitious we get about really removing CO2 from the atmosphere. Because right now, I don't know, carbon. It depends, right? I mean, it's anywhere from a few dollars a ton. I think in the European Union, some of their carbon markets are up to maybe under $100 a ton, but maybe over $50 a ton. And that comes back to the iron story in a way, because so it happened once people started doing these experiments showing iron was limiting. There was at least one group that was interested in then going out and adding iron on a commercial basis and selling carbon credits. This was back in the early 2000s. And when that notion came up and, you know, sort of was being popularized, the oceanographic community really pushed back and said, wait a second, we don't know enough about what the implications are, you know, for the whole ecosystem, for a whole bunch of different other processes in the ocean, of what this iron fertilization would actually do. And so there was pretty strong pushback in the oceanographic community. And then there was an event, I think in 2012, a fellow convinced the Haida tribe, an indigenous group in British Columbia who rely on salmon. They live on some of the salmon rivers up in British Columbia, that he could dump iron off the coast of British Columbia and feed phytoplankton, which would then increase their fish catches. And so he went out and dumped 100 tons of iron off the coast of British Columbia. And there was already been this pushback, and people were concerned about this. And this was, you know, didn't get anybody's permission or anything, just went out and did it.
Did it work? Did the salmon catches increase?
Well, that's interesting. So he created a phytoplankton bloom the size of Connecticut. And apparently the fish, they actually did see a big jump in salmon like the next year, which didn't really make sense, because I think the life cycle of the salmon is like 5 or 6 years. Well,
it could have been younger salmon, I guess.
I don't know.
I don't know, like, the population dynamics of salmon, but but there was no science involved in that, right? He just went out and did this. Didn't really measure anything. People could see the phytoplankton bloom because you could measure it from space, from satellites. But that created a huge turmoil, because it was really kind of illegal. But yes, these sort of treaties, law of the sea is one that sort of governs marine, you know, the marine environment around the world globally. But, you know, like
the United States has never actually ratified law of the sea. So there are these sort of norms. They're really legal norms. It's not like you're going to go to jail if you go out and do one of these things, because it's not technically, I don't know, I guess I'm not even sure it's technically a law, but it becomes a legal norm, and that's kind of what we need to hold ourselves to, I think, anyway. But anyway, he did this, and that created a big sort of a big media storm. Ultimately led to some amendments to the law of the sea that outlaw ocean dumping of iron. Would be considered an ocean dumping, at least for commercial purposes. And all that together sort of, I mean, to me, it really created this sort of stigma around even doing research in this area for iron fertilization as a carbon dioxide removal method. And it had a really negative impact on the whole field. And I mean, it's changed just in the last year or two. I mean, there's now a group of oceanographers that are coming together and starting to discuss doing these experiments and doing, the research and really trying to understand what's going on and what the potential is for this process. Can it really remove significant amount of carbon dioxide? What are the harmful sides of this going to be?
Sounds like that's a real shame, because it doesn't sound like there were any proven negative aspects of it. Like, yeah, he was kind of being a cowboy and went in and dumped his iron off the side of British Columbia, but maybe it did increase the salmon population. I feel like that's something that should have. It's a shame that wasn't explored and was just immediately just shut down, because that, to me, I mean, very first order looking back on it, I would think, oh, well, the algae increase and then it goes down the food chain, and then you would get an increase in the salmon population. So I would think that would be an environmental good to do something like that. But I'm surprised that the oceanographers institutes are so against doing this, I guess because it's not well enough studied.
Yeah, well, there's good reason that we need to understand more about it. I'm certainly not advocating going out and doing sort of, pirate level ocean dumping out in the ocean. Because, well, I mean, first of all, it's not really clear that it works because of these export questions and how much actually gets exported, and then we know you change the ecosystem. So you change the type of phytoplankton that are there. And that was demonstrated in these other experiments they did. And what the long-term effects of that are, over 5-10 years, we don't really understand. So it gets increasingly complex as you dump iron, as you create these phytoplankton blooms. As I said, they get consumed and turned back into CO2, but that process consumes oxygen. So now you have and I think it's, the greater the phytoplankton bloom you have, the more you're going to deoxygenate the ocean, and especially the interior ocean, which isn't very well connected to the surface. So that's going to lead to places, but the potential to, have these sort of significant oxygen depletion events. That in turn leads to production of things like nitrous oxide, because there are microbes that when there's low oxygen, they'll use nitrogen compounds and produce nitrous oxide, which is a very potent greenhouse gas that then can get released. Methane is another one that when you lower the oxygen levels, you can potentially get more methane production. So there are these, what we refer to sort of as offsets of these gases. So it's not clear how much of an effect that will have. I think the oxygen depletion is pretty clear that, if you did this on a really large scale at very large volumes, you would end up with significant oxygen depletion, which, then that would have negative impacts for, any creatures living in the deeper ocean. There's also some concern about some of these phytoplankton can produce toxins. And that's one that gets a lot of press, but there's never really been much evidence, as far as I know, for that actually happening. And for example, when they did these iron addition experiments, They didn't really see much evidence for sort of toxin production, but that's a good one to play up in the press because everybody's worried about toxins in the ocean. So, but yeah, and then there's more complicated effects, like if you add iron to the ocean where it's limiting, then it's no longer limiting, but then nitrogen may become limiting, or phosphorus, and that water gets transformed for literally around the world. And there's one of the concerns is that if you add iron to one region of the ocean, you'll draw down the nitrogen, and then that gets transferred to another part of the ocean where it's more nitrogen limited and becomes even more nitrogen limited. And those can be areas where there's a lot of fisheries and stuff like that. And so those are all based out of models where people have done modeling, ocean models and stuff like that, and trying to figure out how these sort of long-term nutrient dynamics are going to affect things.
So I do want to come back to the modeling, but I also wanted to just kind of clarify one piece that I've been wondering about. So the issue with CO2 fixation, I guess, is that CO2 is absorbed by the phytoplankton, but then the phytoplankton becomes lysed or metabolized, which then causes the release back of the CO2.
Yeah.
But what if it gets consumed by fish in the ocean, or there's some other way to, I don't know, make it sink, something like that, or if someone just like puts a big tarp on it, and brings it down, I don't know, some kind of like goofy way to ensure that it doesn't get lysed, or maybe some kind of genetic engineering solution there.
You get into this this challenge, right, of too many technical solutions for. I don't think that's. The interesting thing about that partly is that when you add iron, you tend to get larger phytoplankton, which will sink a little bit faster, and then you get zooplankton, which are little planktonic animals that actually feed. They're the base. They eat the phytoplankton, and then the fish start eating the zooplankton. And they are an important player in all this. And there's other organisms out there that also are important players in that whole carbon cycle. Fish themselves are probably not that important a player, although there is. This gets back to that whole, the salmon, right?
Yeah, because that's what I was saying, is that you have the phytoplankton that get eaten by the zooplankton, which get eaten by some small fish, which eventually gets eaten by the salmon. And I would think that it would have this positive environmental impact.
Well, I
feel like, but especially with the oxygen depletion in these regions, then it's going to have other problems.
Yeah, that's the thing. We just don't understand the system well enough to go out and. We're probably a pretty environmentally conscious community, which maybe gives us a little more pause about going out and doing major ecosystem engineering and things like that, which is geoengineering, which is the term that we typically use for these large-scale carbon dioxide removal processes, or putting stuff up in the atmosphere to block the amount of sunlight coming in. So I think, I think that's probably partly a sort of a little bit of the ethos of the science of people that study the ocean and stuff like that is, I guess it comes back a little bit to the do no harm kind of rationale. But that is, that fisheries is one of the questions that people have is, if you added iron, would you enhance fisheries? And the answer is probably yes in some places, but then that nutrient issue where you're sort of stealing nutrients from one area and then they're not getting transferred to another area, and those areas especially tend to be sort of more towards the equator, more developing world areas, which rely, in a number of places very heavily on their fisheries. So those are the kinds of complicated questions that come up. I mean, it's interesting, though. There's also a theory that in the Southern Ocean, which is the prime. So when we talk about iron limitation in the ocean, one of the largest area that is what we refer to as the Southern Ocean, which is basically the area north of Antarctica. That goes basically, I mean, you have that whole zone of the ocean down there that you have a South America dipping into a little bit and a little bit of Africa. But the whaling industry, there's sort of this cycle with the whales where something interesting happened when we wiped out the whale, essentially most of the large whales, especially in the Southern Ocean, that feed on krill, which are these little shrimp-like organisms that live on zooplankton, that eat the phytoplankton. And, so what would you think if you took away all the whales? What would happen to the krill populations?
Yeah, then the krill population would explode.
Krill population would explode. That didn't really happen. It hasn't really happened very much. And one of the theories is that those whales, they're eating a lot of krill, they come up to the surface and they poop. And in their poop is a lot of iron. And that there's some evidence that there's sort of this fertilizing system going on there where the whales were actually fertilizing, probably through their waste products, enough iron potentially to sort of enhance the production of phytoplankton krill, and then that was their food source. And again, that's not been really experimentally shown very well, but it's kind of another feedback with iron where I think it's very interesting that.
Always a good theory when it has poop in it, I guess.
Yeah.
So these models are not only are pretty important in terms of climate predictions and whatnot, but they also sound very complicated and very hard to make concise and accurate. I mean, coming from the physics world, I kind of okay, this is my beam, this is my magnet, this is the B field, and it still doesn't reproduce well.
Yeah.
So what are the steps to trying to make a strong climate model or ocean model, or even just an ecological model, like what you were saying, with just a simple system of the whale poops iron that creates the phytoplankton?
Yeah, you should have a modeler on sometime. I'm not a modeler. They are. I mean, they have these global circulation models, and the resolution of them is kilometers, and then they have to simplify everything down to as simple a set of factors. And some of those factors are nitrogen concentrations, phosphorus concentrations, how much sunlight, sort of what the productivity is going to be. But they're pretty coarse, and they do, that's for a lot of this work. We do depend on those models, and then they get into these climate models and global climate models, and those they can run them backwards, I guess, and sort of try to recreate what the conditions over the last 150 years that we have pretty good measurements for are. And if you can recreate those, then that gives you some confidence that looking forward, the model has some real validity. But I guess as an experimentalist, I'm always a little skeptical of models. I don't know how you feel about it.
Yeah, I agree, because it sounds to me like one of the major limits in this setup is the models, because no one wants to move forward with doing a process like this until they understand the impact, and they can't understand the impact unless they have a strong model.
Yeah, right.
And
even if you're limited experimentally in what you can actually do to test the model, right. I think that's a little bit where we are now.
Mm-hmm. What about doing like a lake, like a small scale experiment with something like this?
Lakes aren't iron limited. So no, the thing to do is, the experiments they did, in the 1990s and early 2000s were to go out and drop iron off the sides of ships, and I think those experiments could be redone. I think we've learned more about sort of ocean circulation, where the best places to do this might be. Again, the ocean, I mean, it's amazingly, there's all these huge eddies and stuff that move through the ocean. It's amazingly structured, and understanding sort of how all that ocean dynamics works to try to couple that with these experiments is going to be important. And then we have, I mean, so in the paper that I published was to use these biologically produced oxides, which we think have properties that might keep them up in the surface ocean longer and make them more available to the phytoplankton, but also delivering by aircraft. So as an aerosol instead of dropping it as a liquid off the side of a ship. And that's actually how a lot of iron gets delivered to the open ocean, is by dust coming off the continents. And I think that's something that we haven't really modeled much at all, and it has some different implications for how you would sort of distribute it, the concentrations that you'd actually end up with, and those all play into sort of the productivity and the reaction of the organisms and stuff. So.
Yeah. So in these large open ocean zones that have been treated with iron fertilization, aren't they essentially kind of dead zones? Like, is there anything in there, or does it change as you get to deeper levels?
No. So the iron, as I said, the Southern Ocean and parts of there's some of the equatorial ocean off South America, but also there's zones up in the northern Pacific that are iron limited, and they're not dead zones. No, I mean they are actually, I don't know, moderately productive, I guess you could say. The real dead zones are actually sort of for example, what's called the Southern Ocean gyres, which are sort of the central Pacific Ocean, and those become very nitrogen limited, and those areas have very, very low productivity. And so you could go and add nitrogen. The problem with nitrogen is you need to have a string of tankers going out and dropping nitrogen into the water. And
I feel like there's not a lot of solid nitrogen setups or solid nitrogen.
Well, you could use nitrogen fertilizer, right?
Okay,
Same thing you use on fields, but you need a lot of, relative to the amount of iron you need a lot of nitrogen to overcome nitrogen limitation. So.
And so how is this iron processed? Could you just get a steel beam and just throw it in the water? Does it file down? Are there differences in how they're processed that can make a difference in the size of the flume?
Yeah, well, that's something that I think is interesting. I mean, in terms of the research questions, and those are more probably somewhat experimentally tractable even in the laboratory. Mm-hmm. But the way it's been done is you make an iron solution of it's called ferrous sulfate, and you add some acid to it, which makes it soluble, and then they just take a hose off the back of the ship, put it in the prop wash of the ship, and that mixes it around. As I said, you don't need very much. It's an acidified solution, but I mean, you're adding literally a drop for in terms of the ocean. It doesn't have any impact at all. But that's one way, and then you could put a rusting cable or something like that. That's a source of iron. The problem is it sinks pretty quickly. My own interest came out of this partly is in using the iron that's produced by these iron oxidizing bacteria, which is a unique mineral that it's kind of, they basically they're little nano machines in a way, because they spin out these fibers of iron oxide, which have some organic component to them. Because the microbes have the problem that they're forming iron oxides around the cell, which they can then encase themselves in, which would then seal them off from their environment. So they have to have a way of getting rid of that iron. So they've developed a number of clever mechanisms for sort of extruding these iron oxides. And that we know that form of iron is quite available, because the other problem is, so the phytoplankton have to take up this iron from the ocean. And so that's an important process of how they actually access the iron. And so it's interesting is the kind of iron that comes off the continents is basically from dust, especially in the deserts. And it's a very, what we refer to as crystalline. It's a very sort of solid form of iron. It's actually quite refractory. It's quite difficult for the phytoplankton to access. But still, they're able to do it. There's enough iron there. That's why, for example, downwind of the Sahara, which is sort of the central Atlantic, is not iron limited at all, because there's so much dust coming off the Sahara Desert. But we think that, these more available forms of iron could be quite more effective, certainly, than aerosol dust as an iron source, and that then reduces the amount of iron you need. Potentially it makes blooms last a little bit longer. Maybe it lasts a little bit longer up in the water column, things like that. So that, I think that certainly, if we move forward with these ideas, I think those are sort of some of the experimental areas that we could test pretty effectively, starting in the lab and then moving. I mean, the way this should work is to move into progressively larger field experiments before you start doing any kind of sort of large-scale process of actually trying to remove carbon dioxide from the atmosphere.
So just kind of following the path that the iron takes, I guess. It gets put into the water, and then it gets absorbed by the phytoplankton. The phytoplankton converts it into iron oxide.
Well, they use it in their enzymes, things like that. Yeah,
okay.
Their biological processes.
So is the oxygen depletion is from the zooplankton, right? Not from the phytoplankton.
Well, yeah, probably from both. As they sink into the water column, they're providing a source of organic carbon that's then available to especially bacteria, which will consume a large percentage. Some of it's they can't really get at, but a pretty significant percentage of that carbon the bacteria will consume, and they use oxygen. Or they can use nitrogen if there's oxygen. They'll use nitrate if oxygen's not available. So that is where you start getting into this oxygen limitation question.
So what happens to the oxygen that results from the photosynthesis of the phytoplankton?
So that ends up in the atmosphere.
Yeah. Okay. Yeah.
So about half of, I guess the general number is thrown out is 1 out of every 2 breaths we take of oxygen is produced by phytoplankton in the ocean, and the other is by the trees, plants.
So then how does water get oxygen fixated into it all? Just from like bubbles and... So the
ocean gets oxygenated, well, through fixation, in the water column, and then transfer from the atmosphere, because there's not really any oxygenic photosynthesis that goes on in the deep ocean, and then that oxygen just gets transported through the deep ocean, which is, not much is really happening in the deep ocean, and that's why the oxygen stays there for a long time. It's, so for example, in a lake, right, if you add a bunch of nitrogen or phosphorus, you get these big blooms of algae, which then get consume all the oxygen in the lake, and that's why the lake can go totally almost anoxic, which then kills the fish and stuff like that.
Yeah, I've heard of that happening with like fertilizer and
Yeah.
I imagine there's some kind of differential equation somewhere describing like the rate of oxygen going in and out of the water.
Yeah.
Based on these.
Well, so you've got this whole air-sea gas exchange process going on, which is one process, and then you've got, this chemocline layer in the ocean, which is, this chemical sort of thermal barrier. So you've got processes going along, but and sometimes it really almost comes down to diffusion across those barriers. So you have advection, and then you have diffusion, and advection obviously moves things pretty fast, but diffusion is pretty slow in terms of any kind of larger scale process. So that's where it gets sort of complicated.
I feel like a lot of this is kind of tuning that differential equation to make sure that it doesn't deplete too much oxygen, but you can tune it to eliminate the most amount of CO2.
The math can get pretty intense, I think. I'm not a math. I leave that to you guys. But yeah, I mean, but ultimately you end up sort of a larger scale. Yeah, you end up with these sort of advective processes, which can move things pretty quickly. But then you have these layers where it's almost diffusion across some of those barriers. And again, those are kind of questions that, we have a reasonable handle on, but there's a lot of complexity there.
What happened to the guy who started the company to sell carbon credits? Whatever happened to that company? Is it still limping along, or did it go under, or is it doing well?
Yeah, no. So they've actually, they gave up that idea. Mm-hmm. I think, partly because there was this pushback, and I think, and the whole carbon credit market is
very in flux.
It's in flux is a good way to describe it. And I think that's going to be essential to myself, to sort of making these processes work. And I think, iron fertilization is one. It's not certainly going to be the only one. I mean, even if it works really well, it's not going to pull enough carbon dioxide out of the atmosphere to... Well, if we don't start reducing the amount of CO2 and greenhouse gases that we're emitting, then there's really I would almost argue there's no point in doing this at all, because we're probably just going to screw things up even more. But if we get to the point where we really are, reducing the amount of greenhouse gases that are being emitted, so the IPCC, intergovernmental global panel that sort of looks at this question, right? I mean, they've come to the conclusion that not only do we need to draw down or reduce the amount of greenhouse gases we're emitting, but we are going to actually, to keep temperatures, well, they say 1.5, I don't know. Most people think it's at least 2 degrees of warming by 2100. We're going to have to actually actively remove CO2 as well as reduce the amount of greenhouse gases, and on the order of gigatons per year, which is a real challenge. I mean, I think if iron fertilization worked really well, you might get up to maybe a gigaton, maybe, I don't know, sort of in that range, and so you'd need. But, that's significant, but you'd need probably a half a dozen other methods of CO2 removal to get close to what sort of is the projection is what's required for keeping global climate, sort of under control.
But, comparing this to something like planting forests and whatnot, do you think it has a better shot, or do you think it's on equal footing?
Yeah, I guess I'm not sure I'd know how to answer that question. I think planting forests is good. I think we need a number of solutions. So another one that we've gotten interested in, and there's been quite a bit of interest in, and this is, you talk about sort of the hype cycle, right? Where everybody's excited about something when they start out and then start realizing the challenge. One is, I don't know if you've heard of this, but growing large amounts of seaweed sort of in the open ocean, and then this gets back to your tarp idea, and then sinking it to the bottom of the ocean. And so that's an idea that people have been talking to. It's been quite a bit of interest generated in the last 2 or 3 years. We've been looking a little bit at that just in Maine, because we have a pretty active aquaculture industry there that's growing seaweeds for human consumption and animal consumption. And so can those seaweed farmers, can they actually claim carbon credits? Because some of that seaweed, parts of it they don't, parts of it they just throw back into the water because it's not really for consumption. And so can they claim carbon credits for that? Because that, if it ends up buried in the sediment, then that carbon gets stored away for long periods of time. And so we're trying to assess whether you can use genetic methods to track kelp, in this case, in the sediments to see how much is potentially being stored. And I think, those are all important steps, right? I mean, that's not going to be a global solution, but it can contribute to a global solution. Like then going out and growing, rafts of seaweed the size of Rhode Island and in the middle of the Atlantic and then sinking to the bottom of the ocean. I'm skeptical of that. I think they should do the research. I think people are putting some money into it and trying to develop some ideas around it. But I think it has all the same problems and maybe even more than going out and adding iron to the ocean and fertilizing the natural phytoplankton that are already there. So yeah, I think we need to be doing research at least on a lot of these different methods. And then there's things like direct air capture, which is a much more technological solution, which I think has promise. It's going to require a lot of understanding of sort of more of the physics and chemistry of how you capture CO2 from the atmosphere. But exciting things to work on in science. So.
Yeah, so I mean, there are several kind of scientific revolutions going on in tandem to what you're talking about now. One of them that I like to talk to people a lot because it's so wide-reaching is machine learning, right? So do you think machine learning, for example, is going to be able to try and help some of these modeling problems? Because, I mean, it seems like the perfect fit, this multivariate system that is very complicated and hard to understand with just an analytical model.
Again, I have very little expertise in that area, but there certainly is a lot of interest in machine learning, AI kinds of approaches. Mm-hmm. And I think those are certainly they're certainly coming into our world. As a microbial ecologist, that's sort of these random forest models and things like that I know very little about. My postdocs and graduate students are talking about them as exciting new things. But yeah, as the science develops, and I think, a lot of that will come out of physics and stuff like that, right? And that's really important that knowledge those approaches get transferred. And so, yeah, I think those will definitely help.
What about the kind of genetic engineering revolution that's occurred with CRISPR and now with things like Yamanaka factors and things like that? Do you think it would be possible to maybe create genetically altered plankton to fit these situations better? Like maybe plankton that doesn't require iron for its synthesis, if that's even possible.
That would be a hard one, yeah. I think we're probably, I'm not sure it'll ever be possible to genetically modify a typical open ocean marine phytoplankton that is much more efficient at fixing carbon dioxide and maybe makes big fat cells that sink very rapidly. I mean, that would be the ideal, right? The history of that is not very good in terms of the microbial ecology, because those organisms generally have are limiting in some other adaptation that's required for them to survive in the real world. A good example is nitrogen fixation, which when I was a graduate student back in the 1980s, there was a lot of interest in improving nitrogen fixation because that enzyme actually isn't very efficient, nitrogenase. And rhizobium, which is the bacteria that grows on legumes and actually does the nitrogen fixation process. And so people spent years improving that process, genetically modifying the bacteria, and they could never get them to grow. And they could go out and seed them in the field, and they almost immediately get taken over by the wild. But I think, I do think that in more controlled environments, for example, crops, things like that, developing plants and other, plants, I don't know, trees that could fix more carbon and potentially fix it into sort of more refractory compounds that are harder to break down. That could certainly play a role. And actually getting back to sort of some of these ideas around direct air capture, right? I mean, that's what a plant does, is it's capturing CO2, which is very dilute in the atmosphere. And that's the problem you have with direct air capture technologies, is they've got to work with this very, very dilute CO2 concentration. Whereas if you're trying to capture CO2 out of a coal plant or even a natural gas plant or something like that, the concentrations of CO2 are, hundreds or thousands of times higher than they are in the atmosphere. But plants have obviously, photosynthetic organisms, whether they're microbes or trees, have obviously figured out how to capture that CO2 pretty efficiently. And so I think, potentially coming up with enzymatic processes for doing that, those would be pretty interesting areas to look into.
I'm imagining these like big vats of algae that could be completely well controlled to just do carbon capture.
Yeah, the problem, it's so you should think about that vat, but then you got to think about how much carbon you actually have to capture and how many of those vats you need.
Right.
That's where it gets.
And how much carbon is going into producing all of those vats and
exactly.
running the pumps to make sure it's sufficiently oxygenated.
Yeah. We're starting to look at that more.
Mm-hmm.
And I think that's been a good thing. We're starting sort of this life cycle analyses where you sort of try to really capture the whole process.
So, in addition to the iron fertilization, you also do have a bunch of other projects, like the Coast Cow Consumer Project was one of them that I found.
Yeah.
And MainEDNA was another one that I found. What are some of the ones that stick out to you?
The Coast Cow Consumer, so actually talking about life cycle analysis, there the idea, one of my colleagues, Nicole Price at Bigelow, is heading up a project funded by the USDA, which is kind of interesting. We're a marine science organization, but we get funding from the U.S. Department of Agriculture because they're actually becoming more interested in aquaculture, and especially in seaweed aquaculture and things like that. But it's sort of looking at the ability to sort of couple some of the marine aquaculture to agricultural processes, whether it's animal feeds, potentially using algae or kelp and seaweeds and stuff like that as fertilizers, but especially as supplements for animal feed. And one component of that is it's known that there's certain types of seaweeds that actually suppress methane production in cows, in dairy cows, beef cows, very effectively. And we've actually been looking at that as well, and sort of we have a reasonably good understanding of the mechanism of that, and trying to come up with some plant derivatives that you could use as feed supplements. Because the problem with growing seaweed is that, again, it comes back to that vat, right, as your seaweed, and then how much of that you actually need to actually feed all the cows in America. And those become very large numbers. And
molecularly, how does that work? Because I'm just kind of picturing like cow tums at this point.
So cows have a group of microbes called methanogens that produce methane.
Okay. And
they've got some enzymes in them that are sort of key enzymes in that process of actually making methane. And there's inhibitors. And one of the compounds that the seaweed produces is an inhibitor of that sort of enzymology, one of the enzymes that's involved in making methane. It's quite potent, and it's actually surprisingly potent. You don't need very much of it, and it sort of inhibits that methane production. And so we're interested in sort of whether you can develop that through, including even genetic engineering, to try to develop basically feed additives. Because as I said, the challenge if you actually grow the seaweed is how much seaweed you need to actually add to all the cows. But if you can have a little pill that you can make, then you can potentially make a nutritional supplement that's scalable to sort of a much larger scale. And so we're sort of, I guess we're doing both things. I mean, it's kind of interesting because the coast to cow to consumer really has implications sort of regionally and locally in Maine and New England, and both for the land agriculture side, but also for the aquaculture industry, of developing sort of new markets for that industry, which we'd like to see.
It looks like this effort actually got a little bit more philanthropic support than the iron fertilization has. Do you think that's just because it's a little bit more understood compared to iron fertilization, or do you think there's something else going on?
Yeah, I'm not sure I'd say it was better understood. Maybe it is a little bit. I think it's more acceptable in some respects. I think, some of the philanthropy is, some of it's based around, that it is sort of local and regional, and there are philanthropists that like supporting, sort of the local regional aspects. And in Maine, our sort of coastal ecosystem is sort of, it's big in Maine, right? We have a 3,000-mile coastline when you stretch it all out, and it's sort of central to our economy. And it's kind of a nice economy. It is pretty local, right? We don't have huge sort of factory fisheries or aquaculture or agriculture. It's much more on a local scale, and I think there's value to that. And I think that's some of what drives the philanthropy is sort of maintaining that sort of main ecosystem, if you will. But I think, and there is potential for this to be significant in terms of having an impact, and financially too. I mean, I think we're going to see more resources going into sort of these CO2 mitigation technologies and stuff.
Yeah, it's my understanding that agriculture is one of the largest sources of greenhouse gases. Is that accurate?
I wouldn't say it's one of the largest. It's a larger source. I forget what the exact numbers are. I mean, ruminant. So a significant part of the agricultural greenhouse gas emissions is coming from methane, because methane, again, is a couple hundred times more potent a greenhouse gas than CO2. So if you can reduce that methane emission, then that has a significant impact. I mean, from the farmer's perspective, because we don't actually regulate methane, most farmers, they're aware of climate change and all this stuff, and they would like to see things done about it, but they're also mostly aware of their bottom line, and it's a pretty thin bottom line. Right. And so unless there's some sort of financial incentive for them to reduce having an anti-methane or reducing their greenhouse gas emissions from that perspective, it's a harder sell. But the other thing is, so methane produced by the cow, it's not really totally understood why cows produce so much methane, because it doesn't really do much for the animal as far as we can tell. And so actually, if you reduce the amount of methane, it's basically wasted energy for the animal. So if you reduce the amount of methane energy that's going to that methane production, then more energy actually goes to the animal itself, which can increase milk production and beef production, things like that.
Yeah, I feel like you run into a detection problem again too. Like, how much methane are these cows? You kind of gotta like stick a detector where a cow probably doesn't really want a detector to get this methane.
Well, so most of the methane gets belched out of the mouth, which
Okay.
they still don't like detectors on their faces. But actually, yeah, we have pretty good detectors for methane, and there are, yeah, that's actually, I think there's actually research being done on developing little methane detectors you can put on a cow and have it's walking around in the field and seeing how much methane it's producing. But those are, it gets back, so that actually gets into a verification question, which becomes very important for whatever method, whether it's reducing methane or adding iron to the ocean, is how do you verify how much CO2 you're actually capturing or how much methane you're reducing. And that's a whole nother area that I think that area there's probably more active research going on in now, which is encouraging to see.
I feel like it would be hard to find a very cooperative farmer that was willing to have these methane detectors on their cows.
Well, there's a lot of agricultural research farms that are willing to do that. Yeah, but yeah, the farmers themselves, you have to convince them that there's something in it for them, and that's fair enough, right? 'Cause
do you have any other ideas on how you think agriculture could be improved?
No.
No? Okay.
I'm a marine microbiologist. Yeah, it's sort of funny at Bigelow because we sort of joke because, we're a small marine lab. We don't have any cows at all, but we've gotten into this field where we're actually, but we collaborate with different institutions that have, University of New Hampshire, University of Vermont, for example, have research dairy herds and other research dairy herds and stuff like that we're sort of collaborating with. So I've learned a lot more about ruminant microbiology in the last couple of years than I used to know.
So what is the MainEDNA Project?
So that's a project where we're interested. So eDNA is environmental DNA,
Okay.
and it's the concept that most all living organisms shed DNA into the environment. And so in the ocean, you can go out and collect a liter of seawater, run it onto a filter, and then you'll use a very small filter, and you'll capture the DNA, or a lot of the DNA that's in that seawater. Then now we have the technologies where we can go out and sequence that and then compare it to a database and sort of see what was in that water, whether it was, and most of it's microbes. And we've been using sort of the same technology for microbes for a long time, for the last 30 years at least. But now it's becoming more integrated with higher organisms, with fish, invertebrates, seaweeds. Anything that lives in the ocean is shedding DNA, marine mammals, sharks. And so it turns out that you actually can detect that. And so you can, and it's relatively short-lived, which is good because it means the organisms were probably there fairly recently. That was one of the big questions is, well, was that DNA shed a year ago, or a month ago, or a few weeks ago? So it's becoming quite an effective way to sort of do censuses of what organisms are in a body of water. And we think we can also make it reasonably quantitative, so you get some estimate. That's sort of the challenge, is whether you can estimate how much sort of biomass was there. And so we're interested in that partly sort of from the coastal ecosystem perspective in Maine, is because there's a lot of effort goes into sort of how we manage our coastal ecosystem, and this could be a very valuable tool for doing fisheries assessments, looking for invasive species, watching species change their range, and partly in response to climate change. We're seeing warming of the Gulf of Maine is a pretty rapidly warming body of water. For example, these kelp forests that we're interested in growing this kelp as a seaweed additive, we're seeing those kelp forests move along the coast, change their habitat range in response to sort of the rising temperature, because they're quite temperature sensitive. And so sort of monitoring those changes using eDNA to see what organisms are living, using those kelp forests, for example, as refuge. Looking at things like harmful algal blooms, which is, again, sort of an increasing problem. Again, probably linked to a significant extent to climate change. You get these harmful algal blooms that produce toxins that shut down shellfish beds or can be toxic to fish, things like that. So using these DNA-based methods, you can sort of monitor those, and ultimately we're interested in if we get enough information, can we actually then start forecasting when these processes might happen, to sort of better understand when they're going to happen, and maybe do some remediation of the problems.
Is there any way to use this to look at like genetic drift of the species over time as well?
Yeah, so that's a good question. So what's referred to as population genetics, where you actually look at the a particular species of fish. So normally the way you do that is you get the fish and you take a biopsy of it or something from all the different fish and then compare their DNA to see what populations. But there is evidence that just from capturing that DNA from the water, you can get some sense of what populations are there. Yeah, and genetic drift, things like that, those kinds of processes. Those are sort of, I guess, some of the more cutting edge areas. And then even looking at things like RNA, which is basically the molecules that are being showing what the expression of genes are in an organism. And so if you can actually capture RNA, then you could potentially look at things like stress levels. There's other components, DNA methylation. You can potentially look at age. So those are sort of cutting edge areas, I'd say, in this area. Right now we're good at presence absence. I think we're getting a handle a little bit on quantitation, but then can you actually extend it into looking at things like different age classes or genetic drift, different populations, things like that. It's really, it's pretty powerful.
It sounds like a very useful tool.
Yeah, I mean, that's kind of what it is a tool. But one of the things I like about it is that I can talk to fisheries people now, and, we sort of are talking a common language in terms of the eDNA part of it, because, we're using the same technologies that we're using with microbes. So that, I think, I like that aspect of it, I think it helps to break down some barriers in the science.
So kind of related to this, I saw that you were also trying to have a project that connected the genome to the phenome, this idea of trying to get traits directly from the actual genetic sequence.
So that's a project that's being led by Ramona Stepanauskas, who's another scientist at Bigelow who's been doing single-cell genomics with microbes for quite a long time, sort of environmental single-cell genomics. And single-cell genomics has become a very hot area in human biomedicine. I mean, they now can sequence every cancer cell and stuff like that. But Ramonis has been doing that in the ocean, where they're capturing phytoplankton or bacterial plankton, bacteria, and sequencing their individual genomes. And that's told us a lot about the diversity and how little we actually understand about the diversity. But then trying to tie that diversity to actual function of an individual microbe is that's sort of the next challenge.
And it's a big one from my understanding.
Because you've got to have some kind of an assay that you can link back to an individual bacterial cell, so it has to be very sensitive. And we have ways of separating individual cells by using flow cytometry, which uses fluorescence and lasers as a way of interrogating the individual cell and how fluorescent it is. So if you have a probe that fluoresces in proportion to some reaction that the cell is doing, then you can couple that activity of an individual cell. And then if you can capture that cell and sequence its genome, then you know who it is and sort of more about its metabolic capacity. And so that's sort of what we've been trying to do with that project. And postdoc who's working on that. And a lot of that is about calibration of using laboratory cultures to sort of understand how much fluorescence in this case you get to respiration or some specific activity, so that then you can go to the environment and sort of make estimates about that. So yeah, that's very it's pretty exciting in terms of sort of being cutting edge understanding of coupling that sort of genome to the actual physiology of the organisms.
Yeah, it sounds like a much more experimental approach to kind of like what Google DeepMind has been working on with you get a sequence, right? And then that sequence produces a protein. The protein folds in some unpredictable way that apparently AlphaFold has figured out or something. And then you see how all those proteins interact in the transcriptome to actually get this like macroscopic behavior. Yeah. But if you're just kind of looking at it experimentally, I feel like these two could work super well together to be able to have like an experimental verification of this computational modeling
that's
process.
Probably, I think that's it. Yeah, I think you're looking at something that's, that's the ultimate goal of this probably.
Right, but I'm saying you have this like experimental evidence that you can use as this test bed for a complex modeling process like that. Yeah, which is probably not done in a lot of places.
No, I think coupling in, that's been pretty exciting seeing what's happening with this AlphaFold, because I think that's going to be sort of a new area of being, because in the environmental world, I mean, we can now sequence DNA pretty readily from the environment and do, pretty complete genomes of organisms we will never grow in the laboratory because they're too difficult or, for various reasons. But now, because you can sequence the genome, but now if you can actually predict what those proteins are doing, that's still the big challenge, right? Is knowing what genes are expressed and then what they're acting. And a lot of proteins we don't even know what they are, right? I mean, still, even in E. coli, there's still a pretty high percentage of genes that we don't really even know what their functions are. But if you can now sort of take it to that next functional level by understanding how those proteins are put together, that will be pretty. It's going to be an exciting new area, I think. We're not quite there yet, but
oh no, I didn't think so. I just thought it was good that you guys were, I feel like, developing this kind of codex that can then.
I think that's ultimately where this that's sort of one of the areas I think we see where this could be going.
The last project that I saw was using bacteria to produce nanomaterials.
Yeah, so that was some funding we had from the Navy, and that gets back to the iron and these bacteria that produce these iron minerals, which are they're quite highly reactive. They've got extremely high surface areas because they're literally they're a few nanometer thick fibers, essentially. They're composed of iron oxide, but then also some organic components, which we don't really have a very good handle on at this point. We think they're sort of some type of sugar molecule. But the idea is that those... so iron oxides bind a lot of other things, like phosphorus is one, other metals. And if you can control that process, if you can control the growth of the microbes or even potentially genetically manipulate them in a way to control that process of iron oxide production or even incorporate other metals and stuff like that, can you come up with sort of novel materials? And the bacteria, that's what they do, is make these nanomaterials. So that's kind of the driving idea behind that. We've had some funding from the Navy the Navy's interesting. They'll fund some really pie in the sky kinds of ideas. And so that's where we've sort of been curious about those processes. And again, we're at this point of just being able to grow these organisms in the laboratory is a challenge, and then sort of understanding what laboratory conditions we can change to potentially promote production and stuff like that. sort of where we're at with that.
So another thing I always like to ask is how you got to becoming the scientist that you are now. Like, did you have some kind of formative experience in your youth that was like, I want to do molecular biology and oceanography? Did you like grow up by the ocean or something? I'm curious.
I did grow up by the ocean.
Okay.
Yeah, I don't know. I guess I was just always curious about the ocean. And I did spend a lot of time staring into tide pools as a kid. And yeah, I sort of, I don't think I probably wasn't the best student in high school. And in college, I got more motivated and had some good professors in college that helped motivate me. And yeah, but I didn't actually do microbiology in college. I went to College of the Atlantic, which is a small school in Maine that only has 1 degree in human ecology, and I was sort of doing environmental science in that. But I got interested in sort of ecosystems and how they worked, and thought that microbes were really what are driving most ecosystems, and so I ended up going into microbiology, which I'm happy I did. I mean, I'm working on all these different projects, but they're all tied together by microbes. So it's been fruitful in that respect. But my research is definitely, in the last 5 years, it's definitely gone more towards sort of, I guess, solutions-based research, especially, things like iron fertilization and anti-methane production, those kinds of things. It's really, it's about trying to come up with some solutions to problems versus the real basic science, which the stuff I study with iron bacteria is that's sort of my real basic science interest.
So what do you think are kind of the biggest problems that are in your field today?
Well, in terms of carbon dioxide removal and stuff like that, I think it's just recognizing the problem and the need to do more research and development around it. Certainly with iron fertilization, it's very frustrating, right? That there's no. So now, the money that's coming into that is coming through philanthropy. I mean, we have a small grant from the Grantham Foundation, which is a philanthropic group, to do some sort of this cost modeling for iron fertilization. But I can't write a grant. I couldn't. Well, maybe I think this will change, but I couldn't really write a grant to the National Science Foundation right now and say, I want to study the effects of biogenic iron oxide on carbon dioxide uptake for carbon dioxide removal from the ocean.
I don't know. That seems crazy to me, that NSF sticks their noses up at an idea like that.
Well, it comes back a little bit, to the political situation we're in, right? Yeah, I think there's progress in that, but it's slower than it probably needs to be. And I think, there's definitely more movement, and I think as people sort of recognize that, but yeah, the federal government is being, yeah, I guess for the political situation, it's hard for them to do that directly, and so you sort of have to read the tea leaves a little bit and see where you can get funding for those kinds of things. But it's pretty frustrating, really, because there's a lot of great science that can be done doing that. And, with iron fertilization, the worst case situation from a scientist's point of view is that we're going to find it's not going to work, but we're still going to learn a hell of a lot about the ocean.
Right. Which is going to be important in any kind of modeling.
Which is going to be important for, a lot of other reasons. So
I have my own beefs with NSF. I won't go into that on this.
Yeah, well, we all
we've
all been there.
Yeah. So if you did have a large amount of funding and DARPA or something, a real money group came to you and said, "Okay, I want you to redistribute this funding in order to solve these problems," how would you do it?
Jeez. 30 seconds or less.
Go. You have to decide right now. They're outside that door.
Yeah, I mean, those are, I think, again, you know, coming back to sort of trying to come up with some logical, systematic ways of developing the R&D around these questions is its own big challenge. And I think that's really important. Because I mean, getting back to CDR, carbon dioxide removal, none of these technologies are really, well, that's, yeah, almost none of them are really at the point where you can do sort of commercial level applications, and they're all going to require quite a bit more R&D, and the more logically we can do that, as a scientist, I feel that's the best approach. And so I think, you know, if you had putting the money into that component part of it is really important. I guess my concern a little bit is that the commercial side is going to get ahead of, you know, we're going to put $100 a ton price on carbon, and then, you know, you're going to have sort of this Wild West mentality, right? Everybody's going to be going out and spinning up these ideas for dumping huge amounts of iron or growing huge amounts of seaweed or planting trees all through the Sahara Desert or whatever, and developing huge air capture methodologies that, you know, we don't know really how well are going to work. So I think that's my own opinion in terms of how we should sort of be taking as logical approach as we can, because we know quite a bit about some of the basic science behind these things, and it's really just what's going to be the most effective way of applying them, and what sort of combination do you need to apply them to be most effective.
So if you were, say, talking to a graduate student who's starting out in this field, how would you suggest that they navigate it to try and attack these problems?
Yeah, I think always getting, you know, the best intellectual training you can is the most important thing. Being able to really dig into a problem at a pretty deep level. I mean, that's what's to me the value of graduate school, is that I could really, you know, become the expert in the project that I was looking at. And you know, that's a unique opportunity when you're a graduate student to do that, because then you become a postdoc, and then you have expertise, and you can, I mean, assuming you go that route, that you can really sort of dig into a problem more on your own. But then once you get beyond that level, you're going to have all these, like, I mean, you know, you just listed off all these projects that I'm involved in. My deep level of expertise in most of these is pretty shallow, to be honest, because you rely on, you know, graduate students, postdocs, technicians, you know, people that can really actually get the work done. And for graduate students and postdocs, yeah, it's that intellectual stimulation, right? That's kind of should be to me the driving force, and sort of. But yeah, there's a lot of exciting problems out there.
So what's your team like? Your group of
my team, yeah, it's kind of my actual team that sort of is small, right? I mean, in fact, I have a postdoc who just promoted to a research scientist, so he's pretty much on his own. We work together, and then I've got I co-advise a postdoc, and then we have undergraduates working in the lab. But at Bigelow, and I have a graduate student that I co-advise with somebody from the University of Maine who's working on the eDNA project. But I guess I'm at a point in my career where I'm not trying to hire a huge team. So I'm involved in a lot of these projects, and I help manage a number of these projects. For example, our Coast Cow Consumer Project has, I don't know, 8 or 10 people working on it in different labs at Bigelow. And so, yeah, I guess, you know, my own individual team now is relatively small, but I'm involved. And then this eDNA project has, you know, we have 25 graduate students and several postdocs. they're, distributed among the University of Maine, Bigelow, as well as other institutions in the state. So I'm not day-to-day managing them, but it's sort of a little more overview type management.
Great. Thank you very much for coming on.
Yeah, well,
I
thanks.
enjoyed talking to you
fun.
and learning a lot about carbon capture.
Yeah. Thank you, Lloyd.